The next thing we're going to talk about is a sense of taste. The sense of taste is pretty straightforward. When we have a sense of taste, what happens is we put food into our mouth, we chew that food, and as we chew that food, we break down the molecular bonds that hold that food together. And as we do that, some of the chemicals from those molecular bonds are released. Each food is composed of a unique set of chemical bonds and chemical composition, and those chemicals can bind to the receptors on our tongue, and by doing so, they're going to signal a specific taste. So those chemical receptors are taste buds. Taste buds are found all over your tongue. They're found on your cheeks, on your lips, even on the roof of your mouth. And the way that they act is when that chemical from the food is put into the saliva in your mouth, then it can interact with the taste bud. When it binds, it causes a depolarization and an action potential, and that message is carried to the central nervous system, specifically the taste integration center in the gray matter of the brain, and you can perceive what you're tasting. We have 5 different main types of taste. We have sweet, salt, bitter, umami, and what's the other one? Savory, and sour. So those 5 tastes, we don't have just 5 different receptors. We have, again, hundreds of thousands, maybe millions of different kinds of receptors because they all are going to respond to different chemicals. This is why we don't only have 5 different tastes. You have steak will taste different than chicken, and chicken will taste different than pork. And a chocolate chip cookie will taste different than a snickerdoodle cookie. And it's because they're all made up of those same 5 tastes, but different chemicals and different compositions of those tastes will stimulate a lot of different kinds of those receptors. And each one of those receptors will have an action potential. That action potential will then travel to that taste perception area in the gray matter of the brain, and it will be integrated and put together until you get a complete picture of the taste of the food you have in your mouth. Interestingly enough, most of what we taste isn't really based on our taste receptors in our mouth. It has a lot to do with our sense of smell. Did anyone, when they were kids, someone would make you eat something and you would hold your nose and eat it and protest and kind of like, no, I don't want to eat this, I'm going to hold my nose. And what you find is that works. It doesn't taste as strong if you hold your nose. And that's because the sense of smell and the sense of taste are so intertwined. If you don't have that sense of smell, your sense of taste will deaden. And actually, sometimes people have found that people that sometimes undergo reconstructive surgery for their nose, they'll damage their smell receptors. And if you lose that sense of smell, a lot of people really have a decreased appetite and sometimes struggle to eat because they can't really taste the food that they're eating. This shows the taste receptors. Again, they're located all around your tongue. You have these papillae with the taste buds around them. The taste buds have this taste pore. This is where the chemicals suspended in the saliva will enter. They act on these cells and stimulate these receptor nerves, and then this would be your taste nerve that goes to the taste center that's located in the brain. Next, we look at the smell receptors. The smell receptors are olfactory cells. Those olfactory cells are going to respond to chemicals that are in the air. So before you can smell something, that chemical that you're smelling has to be in the air and it has to be breathed into the nose. Once it enters the nose, these chemicals are going to stimulate these neurons in the olfactory epithelium. These olfactory neurons are going to depolarize and create action potential, and then send that information up into the olfactory bulb. And then from that olfactory bulb, that sensory information goes to the olfactory region in the brain where that sense of smell is interpreted. So it all starts out with a chemical that binds to a receptor, and by binding to the receptor, an action potential is created. Any question on smell? Next, we're going to look at the anatomy of the eye. So the eye is a lot more complicated than your sense of smell or your sense of taste, because you have to think you have a whole organ dedicated to it. Whereas in your nose, you just have a little area of receptors, or in your tongue, you have a little area of receptors. So in order to understand how the eye works, you need to know the anatomy of the eye. There's 3 layers of the eye. The outermost layer is a really fibrous protective layer. It gives structure to the eye, and that's the sclera. So if you ever see the whites of someone's eye, you're looking at the sclera of the eye, and that's the outer region. In the middle, you have the choroid. This is where you have a darker area that has a lot of the blood vessels that will supply the blood and nutrients to the eye. The dark color is really helpful in absorbing any extra light rays that get into the eye to prevent them from giving color confusion or light confusion to the retina. And then finally, the retina is the inner layer. This is a layer that contains the photoreceptors. And the photoreceptors respond to light to give you the perception of vision. The eye also has two compartments. The anterior compartment is in front of the lens. The posterior compartment is behind the lens. The anterior compartment contains a thin aqueous humor. That's just a liquid that's found in the front part of the eye, and that helps with refraction of light. And the reflection of light will help aim it towards the back of the eye. The posterior compartment has a gelatin-like substance that's called vitreous humor, and that's going to help hold that eyeball in its shape, because eyes, just like other areas that we talked about, don't have bones, so there's not a lot to give it that round circular shape. That vitreous humor is the gelatin inside that helps to hold that orb-like shape. Here, when we look at the anatomy of the eye, those things that I just explained to you are a little bit more apparent. The outer portion, this white of the eye, is the retina, and you can see it's on the outside. Part of the retina is the cornea. So your cornea is also part of that outer sclera, or I said retina, sclera layer, and that sclera layer contains the white of the eye and the cornea. The cornea is the first place where light passes into the eye, and we have a refraction of the cornea to help aim that light on the retina in the posterior portion. This is where you would find that aqueous humor. That aqueous humor is just a liquid that's in front of the eye that helps aim the light to the back of the eye. Then we have the middle layer. This is called the choroid, or sometimes people call it the choroid coat. The choroid coat is a lot darker because it does have all of your vascular, so your blood vessels are there. You also have different pigments in that tissue that keeps it dark. This dark layer will help absorb extra light that might not make it to the retina, so that light confusion doesn't occur. It provides nutrients because it does have blood vessels, and it contains what's called the ciliary body, which you can see there, and the iris. The ciliary body is important in allowing for the change in the shape of the lens. So I don't know if you guys know anything about eyesight at all, but being able to focus your eye is based on being able to bend the light, and being able to bend the light to hit the retina at the right angle is based on the shape of the lens. So this ciliary body is going to be able to either contract or relax, because it's actually muscles, and by doing so, it will change the shape of the lens and allow you to refract light to the back of the eye where it needs to be. The third layer is the retina. The retina we find in the posterior compartment only, and this is made up of your photoreceptors. Do you guys know what the photoreceptors are? What they're called? Have you ever heard rods and cones? So those are your photoreceptors. You have your rods and cones, and this layer is just made up of rods and cones that are lined up one after another. The most rods and cones are located right in this area at the back of the eye. We call this the fovea centralis. This is the area where your lens wants to focus most of its light in order to get you a clear field of vision. So when you're wearing glasses, what those glasses do is they redirect, or they bend the light so that it goes and hits that fovea centralis. That's what allows you to have clear vision. The pupil, or the iris, and the pupil on the inside is what allows different amounts of light to come into your eye. Who remembers the eclipse? What was the big deal with the eclipse? What did you not want to do? Why? What could it do? Damage your eye. How would it do that? Too much light, so it burns your retina. It kills the photoreceptors, on the inside of your eye. That's why we have the pupil and the iris, because this is also made up of muscles that can contract and relax to change the size of that pupil. The larger the pupil, the more light is going to come in. The smaller the pupil, less light will come in. So if you're outside and it's the middle of summer and it's really sunny, you'll notice that your pupil constricts, and that's going to prevent a lot of light coming in and damaging your retina. If you're in the dark, your pupils will dilate. So that's how we control that. The other thing you'll notice is in this area, we have the optic nerve that leaves the eye. And the optic nerve is connected to all of the photoreceptors that are in this retina layer. And they all will have their axons collecting in this area and leaving the eye to go to the photo center in the occipital lobe. This spot here, because it's just nerve axons, does not have any photoreceptors. So we call this the blind spot. Every eye has a blind spot, and it's where the optic nerve leaves the eye. And if light is ever trained onto that blind spot, you won't be able to see what's right in front of you. Yeah, so they're detecting light. And they can detect different wavelengths of light. They can detect certain intensity of light. They give the information on the light to the occipital lobe vision center, which interprets that information. So you could think of it as, do you guys know about coding? Zeros and ones, that's what runs a computer. You could think of the rods and cones as collecting those zeros and ones, that information that can run a computer. And then they send all of those zeros and ones, or that code, through the optic nerve to the vision center in the occipital lobe. And the occipital lobe is where you assemble all of those codes together and you get a picture pop up on the screen of your computer. Does that make more sense? Okay. So that's what happens in the eye. You know what that is that's photo bleaching. When something is too bright, especially if you're in dim light, what happens is you expose your rods to bright light and it deactivates them. So have you ever gotten that blind spot when it was really light out? It really only happens in the dark, right? And that's because in the dark, you're using primarily your rods. And your rods are specialized to function in dark situations only. So they're a little bit less clear, but you can see in the dark, which is a good thing. The thing about your rods is if they're exposed to light, it deactivates them. So when you have that flash of light deactivates your rods, your cones can't see in dark light, so you're essentially deactivating the photoreceptors in your eye. That's another thing some people say about pirates wearing an eye patch. So why do you think most pirates wear an eye patch? What could be the benefit? Yeah, part of it was because they were pirates and they would stab each other with swords, and maybe some of them were missing eyes. The other part is it's thought that what they could do is if they keep an eye covered, especially if they're going into a battle that's in a cave or going somewhere where there's going to be a low light situation, is have that eye already just adjusted to the light situation so that when they get in there, because that eye has been kept dark, they can remove that eye patch and that the photoreceptors haven't been desensitized by light, so they have the advantage of being able to see when they were in a lower light situation. Yeah, kind of cool. Yeah, so I guess it's not just for missing eyeballs. Any other questions? So that's just the anatomy of the eye. We'll go through and look at the different parts and explain the function of those different parts of the eye once again. So as I said, the sclera is the white part of the eye. It has the most structural components. So it's made up of fibrous connective tissue. Remember, fibrous connective tissue is a little bit stronger than other connective tissue. So that white part of the eye is going to give the eye a little bit of structure. We also have in there the cornea. So the cornea is still that outer sclera. It's that tough outer part of the eye. If you poke your cornea on accident, You're not going to poke a hole in it. It still has that fibrous tissue, but the cornea tissue is arranged in such a way that it's not white, it's clear. And that's really important because this is the first place where light passes into your eye. This is where you first refract that light. And again, the whole goal of the eye is to refract the light on the appropriate part of the eye, the fovea centralis, so you can get a clear picture of what you're seeing. The pupil is the hole that allows for light to get into the eyeball. So remember, the size of the pupil matters. Too much light, you're going to burn your retina. Not enough light, you're not going to be able to really see what you're looking at. The choroid, that's the middle layer. It's very dark, and that helps absorb excess light. If you had a bunch of light bouncing around in the back of your eye, it'd be really hard to actually put a picture together of what you're looking at. It contains two really important parts. The iris is the colored part of your eye. So this is the structure that regulates size of the pupil. This has pigments inside of it that gives you that color. It's also made up of muscles. So when your pupil is widening or constricting, it's due to muscle contraction or relaxation. And that's in response to the light situation that you're in. And then finally, the ciliary body. The ciliary body is the ring of muscles around the lens of your eye. The lens is the primary area that aims light to the fovea centralis, sometimes called the macula lutea. That's the area in your eye where you want to focus all of the light for the clearest picture. So that ciliary body is really important in changing the shape of the lens so you can have clear vision. The lens is behind, or it separates the anterior and the posterior chamber of the eye, and it's suspended by that ciliary body. So the lens, again, it's flexible, it's transparent, and it's a biconcave structure. We see that that lens is going to change shape in different situations. So if I were to look at something far away and read it, I would see that my lens has a different shape than if it was something that was close to me. We call this visual accommodation. So we need to be able to change the lens shape to be able to see things far away or close up. Accommodation is the term for changing the lens shape so we could read what's close up to us. And interestingly enough, we see that as people age, the ligaments that connect the lens to the ciliary muscle and the strength of that ciliary muscle are going to weaken. So the reason why most older people need glasses is because that ciliary muscle and the ligaments of the lens prevent accommodation from occurring any longer.
So when you have surgery,
so what usually happens is surgery is for near point, or for not near point, it's for nearsightedness or farsightedness. And with that, they change the shape of your lens. So they'll shave off part of it, and that allows for the light to be focused right on the macula lutea. If your lens is too elongated, it's going to have it focus a little bit in front of it, so you won't be able to get a clear picture. We'll look at that in a couple slides. So here we're looking at exactly what I just talked about. We have your eyeball. Here's the ciliary muscle that holds the lens. You can see there's these small little ciliary ligaments. We call them suspensory ligaments. They hold onto the lens. When you're looking at something far away, your lens flattens. And that's because it's going to allow for the light to project closer to the back of your eye. And you can see that it hits right around that macula lutea. If you're looking at something closer, you need that lens to be rounded. So if we're looking at something closer, we see what happens is we're going to have pulling on those suspensory ligaments that pulls this into a round shape. And now we're still focusing that light right on that macula lutea. If this lens was in this same shape here, then we would not be able to focus. Does anyone have any questions?
So like the lens is basically what gives like the eye the glossy look.
I don't know what you mean by the glossy look.
Like, because whenever you look at like eyes, they have like a little bit like glare sometimes. So is the lens what does that?
That's just because you have tears and other liquid always lubricating and protecting your eye. So the water in your eye, the tears, it's a saline solution that gives you that glossy look. Any other questions? The next part of the eye is the retina, and the retina is where we have the photoreceptors. So the photoreceptors are the parts of the eye that actually respond to light. What we see in the photoreceptors is we have light stimulate the photoreceptors that results in an action potential, and then that action potential goes to the visual cortex located in the occipital lobe. The two photoreceptors we have are cones and rods. The rods are less clear, so you get less of a, I guess, clear or crisp picture from the rods. They're really used in low light situations, so when you're seeing in the dark, you use rods. And you might notice that your vision's a little bit more fuzzy in low light situations. They're also usually used in peripheral vision because your rods are not located in as high of a density in your macula lutea. It's usually outside of it. So when you're using your peripheral vision, the reason it's probably pretty fuzzy is because you're stimulating rods more than you're stimulating cones. Cones give you color vision, and they also give you sharper vision. So you're using your cones, which are located in the macula lutea almost 100% of the time when you're in light. When you're in that light, those cones can determine different wavelengths of light, and that gives you the different colors that your body's going to interpret. We can interpret red, green, and blue, and by interpreting those three wavelengths of color, we can get that color vision that we see in. We also have higher acuity from the cones, and that's just because the way that they're set up, they detect light a little bit better. The macula lutea, it's also called the fovea centralis. So this is the area in the eye where we have a very high density of cones. Cones, again, are responsible for color vision. They're responsible for high acuity vision. So this is the area where we want to always focus light in the eye. The other two important parts I wanted to talk about are the optic nerve and the blind spot. The optic nerve is the area that takes the information from the rods and the cones out of the eye. You can see how rods and cones all have axons that leave. These axons are the optic nerve. This optic nerve in this area does not have any rods and cones, so any light that hits the eye right here is considered to be in the blind spot. You won't be able to see something if it's in the blind spot of the eye. Here's a summary of the eye structures. Make sure you read this. This is a good review piece for the test. The last thing I'll talk about is abnormalities in the eye. So the abnormalities in the eyes can be color blindness. If you're colorblind, usually you have either defective rod or defective cones that can't really detect a certain color accurately. Sometimes you don't have those cones, so some people are missing those cones. This is typically found in males rather than females because it is passed down by inheritance in a male-female pattern. Cataracts is when the lens is cloudy. If the lens is cloudy, you could visually see that. I had an old dog with cataracts, and usually that's where you see it more than anything is in older animals. And you could just see that their eye was cloudy. You could tell that they really couldn't see what was going on. And then glaucoma is when you have fluid buildup in the back side of the eye. This could be really dangerous because it can damage those photoreceptors. It can damage the nerves that leave those photoreceptors, and it can cause loss of vision. I mentioned before nearsightedness, farsightedness, and astigmatism slightly. So what nearsightedness, farsightedness, and astigmatism is, it's an inability of the lens to focus light onto the macula lutea or the fovea centralis without assistance. So here we see, if you have a long eye, we call that nearsightedness. You see what's near you. So what we do is we use a concave lens, and that's going to direct that light to the macula lutea so you can interpret that easier. If you have a short eye, you can see that light converges behind the eye, so you need a convex lens to make that light converge sooner. And then when you have an astigmatism, usually the reason for that is your cornea is not even. You might have a bump or an indent in the cornea. So then you have to use an uneven edge lens to accommodate for that astigmatism. Any questions about the eye? Let's take, oh, go ahead.
Are you just multiple eyes?
Yeah, usually. Sometimes if you wear contact lenses too much, so if you sleep in contacts, it could change the shape of your cornea, which happens sometimes, but mostly it's genetic.
That was double. I didn't know if like you want that or go into it.
Yeah, unlucky, I think. Although I do know that there's some kids that are inside a lot when they're younger. Like there's just some kids that don't go outside and they have eye problems too. And it's thought maybe because they don't have to have the muscles changing the shape of their lens, they don't develop that muscle as well. Oh, well, that is unlucky. Any other questions? Why don't we take a 10-minute break and then we'll get back to the ear. So 11:03 we'll start again. And I do have your annotated bibliography and your urinary and respiratory labs up here if you want them back. If you don't want them back, just leave them and I'll throw them out at the end of the day.
Didn't get to your bibliography.
Oh.
Alex, you want yours? Really good job, Alex.
No, I just got them yesterday.
It's a 3-week time frame.
You have a point. As soon as I can, I will.
Just really.
Yeah, I know it's high stress. I'll work on them. I'm hoping today, but either today or tomorrow. We'll see. No? But I want to make sure I read them very thoroughly and give you all the credit where credit is due.
No, yeah, read it. Read mine, read mine.
Yeah.
Read mine. This way is mine.
Okay, does anyone have any questions on the eye before we get into the ear? Nothing?
I have one real quick.
Yes.
How can someone see as a child that something is
So what typically happens is their eye can change its shape, or the cornea can change its shape, and the cornea is that first place that the light goes through. So typically the cornea will change the shape as you age. Any other questions? Okay, now we get to talk about the ear. So the ear is important in hearing and balance. We have 3 different parts: the outer ear, which is generally functioning in gathering sound waves and transmitting it to the middle ear. The middle ear will amplify the sound waves and transmit it to the inner ear. And then the inner ear is where we actually stimulate the mechanical receptors for hearing and the mechanical receptors for equilibrium. The outer ear has 2 different parts. You have the pinna, which is the ear that you see. So this curved area on the outside of your head, and that's the pinna, that's part of the outer ear. And the whole purpose of this structure is to kind of capture sound waves and direct it to your ear canal. The ear canal is where the sound waves will travel to get to the middle ear. Between the outer ear and the middle ear, you have the tympanic membrane. That's your eardrum. That's what those sound waves are going to vibrate in order to get you the sense of hearing. Lining the ear canal, you have different oil secreting structures. You have wax that's produced, and that's to help catch any of the pathogens that might be trying to enter the body. Your middle ear contains that tympanic membrane. the tympanic membrane, you can think of like a drum, and it's a membrane that's vibrated by the auditory sound waves. So those sound waves are going to be transmitted to that membrane. The membrane will vibrate, and then you have the inner ear bones. We call those the ossicles. The bones are the malleus, the incus, and the stapes, and they function in amplifying that sound. So the sound that comes from the eardrum can be amplified by the movement of those three bones. You also have the auditory tube located in the middle ear, and this is the tube that connects the ears to the pharynx. If you've ever popped your ears on an airplane, what you're doing is you're equalizing the pressure inside the pharynx and that middle ear, and that's why you have to pop your ears. That tube is to make sure that the pressure behind the eardrum doesn't get too high, because if it does, it can damage or rupture the eardrum. Yeah, I'm sure it does. It hurts just to pop your ear. I think that's my least favorite part of flying, is I always feel like I've got those ears that are building up pressure so much, and it makes me feel like I'm underwater. I think it hurts. Maybe you're just a tough guy. Yeah. I tried chewing gum. That doesn't always work. Yeah. I don't know. Maybe I have small eustachian tubes. Swallow? Okay. I should write this all down. Usually the best thing that I could do is hold my nose and just blow, and it pops. I don't mind that at all. Okay, so let's look at the picture of the different parts of the ears. This is the outer ear. Here's your pinna. That's the area that's going to direct sound to your auditory canal. Your auditory canal is lined with sweat and earwax. Again, it helps protect the body. It traps pathogens. At the end of that auditory canal, this is our middle ear. That's the tympanic membrane. It's like a drum. We vibrate that membrane, and the vibrations of the membrane are past the ossicles, the malleus, incus, and the stapes. The articulations between them allow for amplification of the vibration. So a little vibration here will turn into a bigger one in the incus and an even larger one in the stapes. And this is where we have interaction with the inner ear. Here we see, this is the auditory tube or eustachian tube. This is where we connect the middle ear into the pharynx. This shows the inner ear. So the inner ear has a couple of different parts that are really important. The cochlea is the area where we get information from sound. So we get information regarding pitch and frequency of that sound wave, and it's transmitted into sound by stimulating little hairs that are nerve hairs inside of the cochlea. This happens as the stapes hits what's called the oval window. As it does that, this whole structure is filled with fluid, and it causes ripples in the fluid that will travel through the cochlea and bend the hairs. The pitch of the sound and the frequency of the sound wave will determine how much the hairs inside are bent, and that will give us our variations that we have in hearing. We also have other areas. This is the vestibule, and this area is important in your sense of gravity and in your sense of your head being forward or backwards, that head position. And then these are your semicircular canals. Your semicircular canals are important in sensing rotation and then acceleration. And it's based on movement of fluid through each of these three different canals that are arranged in all three planes of motion. The information that leaves these areas travels to the either the auditory cortex through the cochlear nerve or to the cerebellum, where body position is incorporated through the vestibular nerve, and they join at the vestibulocochlear nerve. So when we look at the inner ear, we have important areas for both hearing and balance: the cochlea, semicircular canal, and the vestibule. The stapes is one of those middle ear bones that hit that oval window. In doing so, it creates waves in the fluid inside of the inner ear, particularly in the cochlea, the vestibule, and the semicircular canals. The movement of that fluid is going to stimulate nerve endings, which are mechanical receptors, and those mechanoreceptors are going to transmit the information to the central nervous system by creating action potentials. The vestibule functions in gravitational equilibrium. The semicircular canals is rotational equilibrium. The cochlea is going to convert vibrations into nerve impulses. This is a lot of words, so let's just look here. The way that it does that is we have, again, that stapes transmitting vibrations to this whole structure, the semicircular canal, vestibule, and cochlea. This creates waves in the fluid that's located in the hollow tubes. This vestibular canal is going to have fluid move through it, and as fluid moves through it, the structure on top is going to bend down and hit these hair cells. So you can see it a little bit better here. This is called the tectoral membrane, and it can be bent and moved due to the waves in the fluid inside of the ear. When it's moved down, it's going to stimulate the hair cells that are located inside, and when they stimulate the hair cells, those are mechanoreceptors. As you push on them, you create an action potential. That action potential is sent to the central nervous system. In the semicircular canals, we get rotational equilibrium. That's based on the movement of the fluid through each one of those semicircular canals. Each one of them is going to move if you're moving in a certain way and give information based on which one is stimulated. The vestibule is going to respond to gravitation. We can see that head position is altered by that vestibule. So here, semicircular canals, you have 2 of them, 1 or 3 of them, one's in each plane. As you move, you have the liquid inside stimulating those hair cells, and it can tell your body about rotational motion. Same thing with the vestibule. The fluid inside is going to move those hair cells. As it moves those hair cells, you're going to be able to stimulate nerves and then know about your head position. Any questions on this? Okay. So I've gone through this lecture a little bit quickly because I want you guys to have time to read through those labs before this afternoon. We're going to be doing a lot of different labs this afternoon, and if you read it beforehand, then you'll understand it a lot better. So we're going to get out now. You don't have to stay in here and read. You can go wherever you want, but go read through those labs before you come. You probably won't be able to finish them if you don't read through and understand them before we begin.